We'd like to get started, and would like to welcome everyone to this very exciting research rounds at the Heart Institute. It's virtual at the present time, and today we have a special occasion and welcome our Michael Potter, cardiovascular genetics endowed lecturer, and we have the great honor of repatriating a Canadian native, Dr. Benoit Bruneau. But before we introduce the lecturer, I'd like to just say a few words on the Michael Potter family. And this lectureship was established in 2006 through the generosity of Michael Potter. And Michael Potter is a successful Canadian entrepreneur who served for 21 years as the president and the CEO of Cognos. Some of you may know that Cognos is an Ottawa-based leading business intelligence company with a big system data analytics, which was ahead of its time, eventually serving business over 135 countries worldwide. Cognos was ultimately acquired by IBM in 2009. It's still located in Ottawa, forms the leading core component of the artificial intelligence driven decision-making business core of IBM. And Mr. Potter and his family still reside in Ottawa and are proud supporters of the Heart Institute, whose generosity we're most grateful. And Kelsey also mentioned that before I introduced Dr. Bruneau, that for the Endowed lecture today, we have two ways of interacting with Dr. Bruneau. One is that you can use the Q&A feature, which is on the bottom of your screen, in which you can actually post your questions throughout the lecture while it's in progress. And but you are also able to ask live questions today. And so you can use the hand up feature, which you have on your screen. And then you can, Kelsey will unmute your mic and you can ask the question directly to Dr. Bruneau. We'll try that out today. And if you have any technical challenges, please use the chat feature to get things fixed. And I would like to give a particular shout out to Dr. Adolfo Debold, who joined us today. And Dr. Bruneau obtained his PhD working at the Heart Institute with Dr. Debold, and we'll have just that momentarily. So today's Michael Potter lecturer is Dr. Benoit Bruneau. And Dr. Bruneau is the director of the Gladstone Institute of Cardiovascular Disease and professor in the Department of Pediatrics at the UCSF. Gladstone Institutes are world-renowned for the leading edge science, and whose investigators include Shinya Yamanaka, the Nobel Prize winner in medicine for human reprogrammed stem cells in 2012, as a colleague with Dr. Bruneau. And also this year, Jennifer Doudna, and Nobel Prize winner for chemistry in CRISPR-Cas9 genome editing technology, which many of us use. And Dr. Bruneau holds the William Younger Endowed Chair in Cardiovascular Disease and recipient of the Lawrence Florence the George Charitable Trust American Heart Association established investigatorship. And we're most proud of Dr. Bruneau because of his such close connections with the Ottawa Heart Institute when he did a PhD study in physiology with Dr. Adolfo Debold, and including understanding natriuretic peptide. And it's a fitting tribute that together, they just written a state-of-art review on the topic of natriuretic peptides, published in Nature Review Cardiology in the November issue. And Dr. Bruneau then did his postdoctoral fellowship with cardiovascular genetics in the laboratory of John and Cricket Seidman at Harvard. Let's remember, his wonderful presentation at that time on the biology TBX5 and the relationship to Holt-Oram syndrome, published in Cell, just amazing science. And then I was also at the director of the Heart and Stroke Center at U of T, and that was a proud moment when I was able to work with the Hospital of Sick Children, recruit Benoit back as the scientist in the Research Institute and also the Department of Molecular Genetics. And Benoit had an amazing trajectory in terms of science publishing. Nature Cell was amazing discoveries. I remember IRX5 and many others. And but then I was heartbroken when I learned that Benoit is leaving us, going for San Francisco for Gladstone. Oh my God, that's amazing. And that's because his wife visited San Francisco, just fell in love with the city, which many people are proud to. Benoit's research is really aimed at understanding the mechanism by which gene programs that he deployed in cardiac differentiation really is discovered and mapped many of the key functional control regulators, of cardiac development and the implication for congenital heart disease. And now is turning some of these tools in terms of regenerating cells in the setting of heart failure, for example. And he published in top impact journals, as we mentioned earlier, and he just received a prestigious 4D Nucleum Award of $3.6 million from NIH with Katie Pollard at also the Gladstone Institute, and really will help to map, the future of congenital heart disease. And hopefully we'll learn some of this during his lecture today. So we really look forward to the 2020 Michael Potter Endowed Lecture in Cardiovascular Genetics by Dr. Benoit Bruneau, entitled Transcriptional Regulation of Heart Development and Disease. Benoit, take it away.
Oh, Peter, thank you so much for that really too kind introduction. It's really, really an honor to be selected for this lecture series, and I hope I can live up to the expectations. It's been quite a trajectory. I've been thinking I'll show you a bit of a historical slide. I do, especially with Dr. DeBold on the call today, I do want to bring to everyone's attention the impact that the discovery of ANF has made. And other than completely revolutionize how we understand how the heart functions and how it interacts with the other organs, I'll just fast forward to last year when the first real effective new heart failure drug, Entresto, was put on the market by Novartis. And this is built on the discovery that ANF can be protective for heart failure. And this is very personal for me because my father switched to Entresto as soon as it was available. And it's the only thing that's preventing his heart failure from crashing below 30. And so I'm personally grateful for the discovery of ANF, but also, of course, it launched my research career. So thank you for having me on, and I will share my screen and get started. So presumably everyone can see this, and presumably people can see my pointer.
Yes.
So I'll start with this. People on Twitter will know this meme, how it started. And this is me as a graduate student in the lab on the 3rd floor in the Heart Institute with proper PPE, although now I'm told we're supposed to wear gloves when we pipette. That's news to me. And so this is the beginning. This is how I started in the lab. And this is how it's going now. We're wearing masks when we're near one another. And this is the majority of my lab here in the quad at UCSF, celebrating a birthday with some cake and fruit. And it's been an amazing journey of close to a quarter century. And as Peter outlined, there's been many people, many mentors and colleagues along the way who've helped me along, and I'm eternally grateful for everyone who's helped guide me towards where I am now. So I'll tell you about the work in the lab that is aimed at understanding what controls lineage specification. So how does a pluripotent stem cell get committed to a cardiac fate and differentiate into a cardiac myocyte? And how does these cells, using the same genome as an instruction set, get committed instead to becoming neurons? And we're interested in these cell fate events, and we're interested also in how this transitions from developmental plasticity to phenotypic stability as the progression of differentiation takes place. And of course, In heart failure and dramatically in cancers, this phenotypic stability is deregulated, and there's an increased plasticity that accompanies many disease states. And so the biology that we study is gene regulation. So we're interested in understanding basically anything that touches DNA and helps turn on or off genes. So this includes DNA binding transcription factors, chromatin remodeling complexes, histone modifications, and more recently, the 3D interactions in the genome. So the first thing I'm going to tell you about are DNA binding transcription factors. And in particular, as Peter alluded to, one that I've been working on since my postdoc, TBX5. This is a member of the T-box family of transcription factors. In work that I published in the last millennium, I showed that TBX5 is exquisitely restricted to the atria and the left ventricle, but not the right ventricle and the outflow tract. And importantly, for disease and development, TBX5 heterozygous mutations were the first mutations identified to be associated with congenital heart disease. And so here I'm showing you an echocardiogram of a patient with a TBX5 mutation, and you can see that this patient has a muscular ventricular septation defect, an opening between the left and the right ventricles that shouldn't be there. And the two important things that came out of this discovery was that, first of all, congenital heart disease is at its root a defect in developmental biology. And this is perhaps not surprising, but this was cemented by the discovery of this. The other, from a transcription factor point of view, is that these are all heterozygous mutations, and for the most part, they're loss of function mutations, implying that a 50% dosage reduction in a transcriptional regulator is sufficient to impair the normal development, in this case of the heart. And this has been borne out over and over again. So the stoichiometry of transcriptional regulators is very important. And so we've been trying to understand how the reduction in TBX5 dosage results in congenital heart disease and what this means in terms of transcriptional regulation. I'm going to show you just one very brief vignette on what we're doing in the mouse before shifting to human. And so one of the discoveries we made a few years ago is that with genetic labeling, I won't go into the details of how this is done, we're able to genetically trace the precursors of the left side of the interventricular septum. A very, very specific population. You can see here an embryonic day 10.5, that the red fluorescence labels this lineage, which persists later in development, and much later on in development, you still have these cells that populate only the left side of the interventricular septum. From understanding lineage contributions to the heart, this is really important. But what it's allowing us to do is to be able to understand defects in ventricular septation in a very specific way. So here I'm showing you 3D light sheet, so 3D projections of light sheet microscopy, where we've labeled the TBX5 lineage in green, and then this red lineage that I showed you that lines up only on one side of the interventricular septum with a very, very sharp boundary. And so this is what the lineage looks like in a wild type up top. But at the bottom, we have mice in which we've compromised the dosage of TBX5. And as I'll show you in a bit, this results in septation defects. And what you can see right away is you no longer have this sharp alignment of the green cells and the right cells bisecting the junction between the left ventricle and the right ventricle. And so we can take these 3D volumetric imagings, and we can slice through and at various levels image this sharp boundary of red cells abutting lineage negative cells. They're basically like a Roman phalanx, protecting the army behind them. And you can see that in the TBX5 mutant embryos, hearts, that you have this disruption in the alignment of these cells and the junction with the green cells, and even some that have escaped off into the right ventricle where they're not supposed to ever contribute. And you can see here close up where here finally you can see I've outlined the ventricular septation defect, and you can see that at the base of the interventricular septum, whereas normally the wild type hearts are enriched in this lineage, and then lower down you have this sharp alignment again. These cells are less prevalent at the base, and their alignment is disrupted. And so we're using now this 3D volumetric approach to be able to study the cell biology of septal formation and how it goes wrong in congenital heart disease, and as well to be able to use the lineage labeling when in our single cell RNA-seq and ATAC-seq experiments to be able to identify which cells we're looking at, so we can look at defective gene expression. And we've begun doing this, and hopefully in a future lecture you'll be able to hear the fruits of those labor. But I want to go to human heart development. And we have the ability now with the discovery of iPS cells and CRISPR engineering to take iPS cells either from patients or in what we do normally these days is to take a well-known, well-characterized iPS cell line and engineer in a mutation. And then we can use various protocols that have been derived from an understanding of embryology, of lineage commitment and differentiation, and turn them, as you see here, into beating cardiac myocytes. So these were cells that were once someone's skin cells that were turned into iPS cells and then were directed into cardiac differentiation. Now, this is great because we can make tons and tons of human cardiac myocytes and other cell types, but I would suggest that the most astute cardiac anatomist would not be able to tell where the interventricular septum is in this dish of cells, let alone the atrium or the ventricle. So in trying to understand congenital heart disease, can we even understand anything about congenital heart disease, which is a 3D anatomical defect? And what I'm going to tell you is that we can to some degree. And so this is the work of a pediatric cardiac anesthesiologist, Irfan Kateria, along with a team of folks who've worked over many years. And what Irfan and team did was to take this parental cell line, Bruce Conklin's wild-type C11 line, a line that's karyotypically stable, well-characterized, easy to engineer, or relatively easy to engineer these days. And then what they did is to CRISPR edit such that we have a line that's heterozygous for deletion of TBX5. So this will mimic, this is the disease model. We have two different lines that are homozygous for the TBX5 deletion. So this is the complete loss of TBX5. And then we have a control line that has seen the CRISPR machinery, has been passaged the same as the others, but doesn't have an indel in the TBX5 gene. And you can see here below by Western blot that you have two isoforms of TBX5, a long and a short. There's reduced dosage of TBX5 in the heterozygous, and there's an absence of TBX5 protein. So we have this allelic series now of human TBX5 iPS cells that we can use. And so we can use this allelic series, differentiate them across a time course of differentiation, and we use day 6, which is a cardiac precursor, and day 11, which is early cardiac myocytes, or day 23, which is a more mature cardiac myocyte. And what we find when we interrogate the physiology, because obviously there's no anatomy to look at, we interrogate the physiology of the cells either by patch clamp or looking at calcium transients, and we find that in the homozygous TBX5 cardiac myocytes, there's a marked prolongation, sometimes as long as several seconds of the action potential duration, with a modest but not statistically significant alteration in the heterozygous cells. And there are defects in calcium transients that are significant in the heterozygous lines and very pronounced in the homozygous lines. And this is in line with a primary diastolic dysfunction that we described many years ago in both mice and patients with TBX5 mutations. And so this correlates with the physiological defects that patients with congenital heart disease have, irrespective of the structural heart defects that they have. We also looked at sarcomere structure and cell size, and we find that in the heterozygotes, and it's a bit hard to see, but you can see here that there's a degree of sarcomeric disarray that is different than the nice orderly sarcomeres that we see in the control lines. And this is far more pronounced in the homozygous lines. The homozygous cells are also much larger for reasons that we don't understand. And again, this correlates with sort of a cardiomyopathy type feature that one sees occasionally in patients with congenital heart defects. So we have cellular features of the disease in our disease model. So that's reassuring. So we took these differentiated cardiac precursors and cardiac myocytes, used the 10x drop-seq approach to do single cell RNA-seq reasoning that there was two possibilities of how the TBX5 mutation might be manifest. One might be that there are defects in gene regulation across all cardiac myocytes, and that the reason that certain structures in the heart are more affected, for example, the interventricular septum or the AV node, is that those defects in gene regulation have more of an impact in those structures, that those structures are more sensitive. The other potential is that there are only certain subsets of cells within the heart that are sensitive to the dosage of TBX5. And if you took the entire heart, ground it up and looked at gene expression, you may not see subtle defects in, for example, in gene expression in the atrial septum. So we use the single cell RNA-seq, hoping that we may be able to tease out these possibilities in the dish in 2D, albeit an imperfect system. And so here below are UMAP representations of single cell RNA-seq. So each dot is the transcriptome of a single cell. And this is mapped in 2D. This is a highly dimensional data set. There are thousands of cells with each sequenced to a depth that we can measure 50,000 transcripts per cell. So it's a high dimensionality that's been reduced to 2 dimensions. And I'm using a color scheme here that I'm going to use throughout, where black and green are the control cells, red are the disease model, the heterozygous TBX5 mutants, and blue are the homozygous. And what you can see at day 6 is that there are 3 major clusters with 3 other sort of more minor clusters. This cluster down here are the nascent early cardiac myocytes and the cardiac precursors, and these are other types of cardiac precursors. And what you can see is that the genotypes pretty much mix between the clusters. There's no single cluster that's driven by a particular genotype. When we get to day 11, when now there's more differentiated cardiac myocytes, what you can see is, again, many of the clusters are mixed with all the genotypes. It's all salt and pepper. All 4 colors are mixed together, except these clusters up here where you have this black and green cluster, this control cluster. You have a red cluster that's predominantly a heterozygous cluster and a blue cluster that's predominantly a homozygous cluster. So now we're seeing that there are certain subtypes, but not all cells, that are driven by one genotype or another, indicating that the gene signature is disrupted enough because of the change in TBX5 dosage that this is driving the identity of the clusters. And by the time we get to day 23, the predominant driver of clustering is indeed the genotypes. So again, we've got the wild type clusters black down here. Nearby, we have a heterozygous cluster. So the wild types and the heterozygous cells are distinct, but near one another. And we have homozygous clusters that are farther apart, telling us that the genotypes are now driving the phenotypes in a broad way. So what do we learn from this? So let me go back. Again, I've got in the inset here the troponin T positive cells, which are the cardiac myocytes. So we have other cells, we have fibroblasts, endothelial cells in the dish. And so we take the troponin T positive cells, we recluster them. And now again, we have the black and the green cells here, adjacent to the heterozygous cells here. And then out here, we have the predominantly homozygous cells. And here at high resolution, we have around 15 independent clusters that are computationally called as being different from one another in terms of their transcriptome. And on the right here, we can draw basically a family tree that tells us which clusters are similar to one another. Because if we want to ask which wild type cluster do I compare to which heterozygous cluster, for example, you wouldn't know a priori just guessing. So this family tree is telling us that, for example, cluster 1, which is over here, which is predominantly wild type, is more closely related to cluster 2, which is out here, which is mostly heterozygous, and so on and so forth. So we can now do pairwise comparisons, and what do we learn? So now we're looking at cluster 5 versus cluster 7. So cluster 5 is a mix of wild type and heterozygous cells. Cluster 7 is mostly heterozygous cells. And we look and see what are the changes in gene expression. And what I've colored in orange are genes that are related to electrophysiological or arrhythmia phenotypes. So you can see ryanodine receptor, NCX. And in purple are genes that are genetically associated with human congenital heart disease. And what you can see is that there are a number of genes. And so what I'm showing you in the bubbles here indicate the intensity of the bubble is the level of gene expression. and the size of the bubble is the number of cells expressing. So you can see here that titin, for example, is expressed robustly in the wild type cluster, and it's almost absent in the heterozygous cluster, whereas MYL2, ventricular myosin light chain, turns on in the cluster. And so we get this list of genes, and when we look at a different comparison, so again, the wild type cluster 1 versus heterozygous cluster 2, you can see that this set of genes up here is common between the two comparisons. But now there's a completely different set of transcripts that are altered in the heterozygous cells, indicating that indeed there are some commonalities in the response of reduced TBX5 dosage when we look at different cell populations, but there are differences, there are vulnerabilities that are specific to various cell populations. And we can look at this across the different time points to then be able to understand dosage sensitivity. Here I'm showing you now the gradation of gene expression where you're looking at wild type, heterozygous, and homozygous. And you can see here NPPA, which encodes ANF, is very, very sensitive. It's almost completely off in all cells in the heterozygous cells, and more so even in the homozygous cells. Whereas another gene that's involved in sodium currents, TECRL, is partly reduced in the heterozygous cells and then largely off in the homozygote. So there's different graded responses to dosage. And here we've confirmed the ANF gene expression by RNA-scope, where you can see that there's red signal here in the controls, there's faint signal in some of the heterozygotes, and it's off in the homozygotes. So again, telling us that we can uncover both sensitive and also graded responses to TBX5 dosage. Now, of course, this is all in a dish. And so we took advantage, and so we wanted to know, are the genes that we're looking at in a human heart spatially restricted? So we took advantage of a published spatial transcriptomics data set published in Asphodel. So this is not experiments that we did, it's experiments that were published, but the data set is there. And what they did is to take human fetal hearts. It's very low resolution, but they have spotted arrays where they take out the tissue spots and then do RNA-seq on the spots. And you can see here in this cartoon representation in B that they've classified compact ventricle, trabecular ventricle, atrial ventricle, outflow tract, and so on. And so we took our differentially expressed genes and we painted them on. And so you can see here TBX5. So the darker the signal, the higher intensity the expression is. So TBX5 is more intense in the atria and it's lower in the ventricles, just like we'd seen in the mouse. ANF or NPPA is very strongly expressed in the atria and in the in the trabecular myocardium of the left ventricle with some expression in the rest of the ventricle. Again, consistent with what we've seen in the mouse, but interestingly now this is fetal human heart. Here's a surprise, COL2A1, which is upregulated in our TBX5 disease model, is largely atrial specific, and IRX4, which we had shown many years ago, is in the human heart ventricle enriched. And so we took all of our differentially expressed genes, or downregulated genes here, and painted them onto this data set to ask, what's the anatomical distribution? And you can see that there's genes here like GJ5, which encodes connexin 40, is atrial restricted. And so we can say, this gene here, PRKG2, is atrial restricted. And we didn't know this a priori. And we can say that despite the differentiation being in 2 dimensions, we can, based on this spatial transcriptomic data set, say, these genes that are downregulated in our TBX5, our human TBX5 cellular model, are likely to be involved in some atrial physiological or anatomical chain, whereas these genes out here, like IRX4, GJA1, IRX3, would be mostly involved in some aspect of ventricular morphogenesis and physiology. And we could extend this to all of our genes, giving us a more anatomical 3D perspective on the changes in gene expression that we see. Okay, so then what can we learn from, we've got lots of genes, we've got an alphabet soup of genes, what can we learn from it in terms of how these interact with one another? So we collaborated with the lab of Holger Hein at the CRG in Barcelona. They developed an algorithm called Big Scale, which allows the formation of gene regulatory networks, not based on a priori knowledge of interactions like protein-protein interactions or things like that, but simply looking at the Z-score of each excuse me, each individual cell. So this is taking advantage of the single cell RNA-seq and its complexity and correlating every single transcript to each other and looking at not just the relative levels, but the change in gene expression across cell types, looking at the correlation score. And so each dot in these hairballs is a node of centrality, is a node, and the lines are edges that connect genes to one another. And I want to bring your attention, so here in purple, I've color-coded some genes that are linked to human congenital heart disease, and in red are some genes that are involved in heart development. And here you can see there's a SMAD2 representation of a node of centrality. Oh, and by the way, the node of centrality is calculated using the PageRank algorithm. This is the Brin and Page algorithm that was published many years ago that led to the formation of Google, which made all the other search engines oblivious. And what it does is it takes into consideration not just the linkages that you have, but the importance of those linkages within the neighborhood. So the size of the dot here is the increase, is the degree of PageRank centrality of the gene within the network. And so here you can see that SMAD2, which is in the wild type network, is absent from the disease network. And so it's been completely kicked off because of its linkages. MEF2C, an important cardiac transcription factor, is present in the wild type lineage. It's present in the heterozygous gene regulatory network also, but it has reduced centrality. And it has reduced centrality despite being expressed at the same level. So it's its connections and the importance of those connections that have been reduced. And this was further amplified by ranking all of the genes that have TBX5 dependent reduction in PageRank and correlating them with TBX5 expression. And you can see that MEF2C, along with other congenital heart disease associated genes, is right at the top. It highly correlates with TBX5. And so this was very intriguing to us. We know that TBX5 can turn genes on with MEF2C. TBX5 and MEF2C are part of a reprogramming cocktail that can transform cardiac fibroblasts into cardiac myocytes and regenerate hearts. And so we asked whether there'd be a genetic interaction. And so we took advantage of a hypomorphic allele of TBX5 that does not have any defects in heart formation. A heterozygous deletion of MEF2C, which also has normal heart structure. Then when we put the two alleles together, the compound heterozygous of TBX5 and the null allele of MF2C, we now see muscular ventricular septal defect consistently across interactions. This is really unusual in the mouse. There's no good mouse model of muscular VSDs except for the TBX5 null heterozygotes. And so this has uncovered from the genetic network a very sensitive genetic interaction between TBX5 and MEF2C. And we were able to look at chromatin occupancy of TBX5 and MEF2C in mouse hearts and show that there's a very strong correlation of TBX5 and MEF2C occupancy, also of MEF2A, which comes out of the gene regulatory network. So what I've shown you so far in this part of the lecture is that we've been able to start with iPSL modeling of disease, do single cell RNA-seq to identify genes in vulnerable populations that are sensitive to TBX5 dosage, develop gene regulatory networks, and then have testable hypotheses that lead to a new understanding of cardiac morphogenesis and the genetic interactions that take place. And we're continuing this to now understand what the chromatin level basis is of TBX5 haploinsufficiency. So in the last bit, I want to talk to you about chromatin remodeling complexes. These are complexes that are recruited to the genome by the DNA binding transcription factors. They're involved in moving nucleosomes around and organizing the structure of the chromatin. So this is the work of a postdoc in the lab, Sutansu Hota, with help from other colleagues. And Sutansu has been working on a complex that's known as the BAF complex, so the BRG1, BRM associated factor complexes. This is a large multimeric assembly of complexes. It has an ATPase that moves the nucleosomes around, it's the engine, and it can be represented by one or other mutually exclusive subunits, Brahma or Brahma-related gene 1. And the concept here is, for example, you can see BAF60, the BAF60 subunit can be represented by BAF60A, B, or C. And so you can imagine with all the various combinations of all these mutually exclusive subunits, these different variants, you can have tons of different types of flavors, I guess, of complexes that function in the genome. And we got into this a number of years ago when together with Jenna Rossen's lab, we made the discovery that one of these subunits, BAF60C, is highly enriched in the developing heart and in somites. And this is the first example of a tissue-restricted subunit of a complex that was thought to be ubiquitous. And in subsequent work, we showed that BAF60C is important for the pioneering activity of the cardiac transcription factor GATA4 to then bring in the chromatin remodeler to turn on cardiac genes. And so Swatansu set out to understand what's the nature of the chromatin of these complexes across cardiac differentiation. And this up here is really important for me to highlight, which is an in vitro directed differentiation system from mouse ES cells. We then apply, this is from Gordon Keller's lab, we apply VEGF activin A and BMP4 for about up to 4 days. This generates fairly homogeneous mesoderm. And then after that, we transition to a different cocktail of cytokines that then results in about 80% of beating cardiac myocytes at the end of 10 days of differentiation. So this is the system that we use, very, very strict regimen of cytokines to be able to drive this stepwise process. And so Swatansu used a BRG1 triple flag knock-in to IP the complexes. This is the Cipro Ruby stain showing you the reproducibility of the complexes. With Nevin Krogan's lab here at Gladstone, we did IP mass spec and over this time course of differentiation. And you can see that the complexes shift from a flavor that's enriched in pluripotency to a different flavor of complexes that's enriched in cardiac precursors and cardiac myocytes. We see, as we expected, BAF60C, the cardiac specific subunit. We also see what we didn't expect, which is a subunit called BAF170, and other things that we didn't expect, like WDR5, which is normally thought to be part of MLL complexes. Swatansu did the reciprocal experiment, again with a triple flag tag into BAF170, again across the time course of differentiation, did IP mass spec. We get a similar flavor of complexes. We pull out BRG1, of course, and we also pull out Brahma. Now, this is intriguing to us in a sense because sure, we expect Brahma. We expect there to be their BRG1 complexes and Brahma complexes, but Brahma is thought to be dispensable for embryonic development. Brahma knockout mice are born at Mendelian ratios and go on to then sort of develop tumors. BRG1 is absolutely essential for early embryonic development. So what's Brahma doing here? And we know from genetics and from cancer biology that Brahma and BRG1 are redundant with one another. So Tansu went and said, okay, look, CRISPR works well, let's delete Brahma and see what happens. And so I'm showing you here 3 movies, and you can see that as I scroll over them, that the movies are actually running. And here are the wild type cells on the left, they're beating vigorously. And here these are 2 Brahma knockout lines. And if you squint, you can see the Brownian motion of the cellular debris, but these cells are not beating. And yet they're making tons of cells. So what's going on? So we did single cell RNA-seq, and the punchline is that whereas the wild type cells here in red are making largely cardiac myocytes, the knockout cells, the Brahma knockout cells here in blue, are making fibroblasts and osteoblasts in these clusters here, but out in these clusters here, they're making neurons. So as you can see here, they're making beta-3 tubulin positive neurons, they're making retinal type neurons, they're making other types of CNS type neurons. So this is really puzzling to us. What's important is what they're not making. They're not making cells that normally would be the product of an impaired cardiac differentiation, which would be hematopoietic stem cells or endothelial cells, and they're not making endodermal derivatives. So they're making a specific subtype of mesodermal derivatives, but they're mostly making neuroectodermal derivatives. And you can see here by beta-3 tubulin staining that they're in fact making cells that express robust levels of beta-3 tubulin and have these neurite-like extensions and don't express cardiac troponin T. So what in the world is going on? So when do these Brahma cells diverge from cardiogenesis and how? So we used, again, single-cell RNA-seq at various stages. And here I'm showing you a UMAP of a PAGA graph abstraction. And you can see here at day 4 at the mesoderm stage that the Brahma knockouts and wild types cluster together. And so transcriptionally, they're very similar to one another. Whereas as soon as you get to the precursor and cardiac myocyte stage, now the wild type cells take this blue path and end up at cardiac myocytes. And now the knockout cells take this orange path and end up as neurons. And we can look at feature plots where at day 4, the mesoderm cells are all expressing EOMES in a very nice way, whereas the wild types now are expressing troponin T, but the knockouts are expressing neuronal markers like GBX2 and CRAB-P2. So they're very rapidly adopting a neuronal fate from this mesoderm state. So I want to emphasize this. They're making mesoderm just fine, but then they turn into neuronal precursors. And so we can look at this in a different way, where here we're using a trajectory inference algorithm called Erd. Erd is the Norse god of fate. And you can see here that when we look at the root at the day 4, the knockouts, which are in orange, and the wild types, which are in blue, all occupy the same mesoderm state. And the wild types take this stepwise differentiation towards various subtypes of cardiac myocytes and fibroblasts, whereas the knockouts immediately make this transition to various types of neuronal precursors. And again, if we look at markers, you can see here all the cells express MESP1, they're all making mesoderm. The wild types make troponin T positive cardiac myocytes, whereas the knockouts jump and immediately make SOX2 or beta-3 tubulin cells. So it's a rapid jump from supposedly committed mesoderm to now neuronal precursors. So we look to see how is this happening. We look by ATAC-seq to look at chromatin opening. At day 4, the only difference that we see is in sites that are less accessible, and these are sites that are near genes that are involved in signaling or genes that will turn on, that are being primed to be activated, like in the TBX5 gene, which is not on at day 4, but will be on subsequently. And so you can see here, these enhancers of TBX5 are less accessible in the knockouts than they are in the wild types. At day 6, now that the knockout cells are transitioning to neuronal precursors, now we see that cardiogenic genes like NKX2-5 are no longer accessible. This is consistent with what happened at day 4. But now we see this neuronal enhancer of the very potent BHL8 transcription factor ASCL1, which is closed in the wild type, is now open for business in the Brahma knockouts. And this down here is showing you histone modification in neuronal precursors, telling us that this is in fact a bona fide neuronal enhancer for ASCL1. So we're getting the de novo opening of neuronal enhancers. Okay, but that doesn't tell us what's happening at day 4 at the mesoderm stage. And so when we looked at K27 acetylation, what we find at day 4 is that there are some genes where there is a de novo gain of K27 acetylation in some of these loss in these cardiac genes, and a gain in some of these other genes. And when we look at the motifs that are enriched in the peaks that have increased enrichment of this active mark of histones, H3K27 acetylation, we find that the most enriched motifs are those associated with POO factors, OCT4, OTX2, OCT6, BRAIN1. These are all the same family of transcription factors that bind these shared motifs. And so we looked at a variety of these different candidates, and what we found was that OCT6 in particular, which is expressed lowly at day 0, cranks up at day 2, and in a wild type would be shut down, lowered at the mesoderm stage and completely shut off by the time you get to a cardiac precursor stage, and it's maintained at the protein level in the Brahma knockout. OCT6 is a neurogenic transcription factor and can de novo activate neuronal genes. So we asked whether this would be one of the initiating events. And so Tansu did POO3F1 or OCT6 siRNA in the knockouts, and whereas this does not rescue the cardiac differentiation, it prevents or largely prevents the presence of this beta-3 tubulin staining, indicating that at the very least for the neuronal induction part of things, it's this sustained expression of POO3F1 or OCT6 that's important for driving this neuronal differentiation. And now when we did Brahma ChIP-seq, and when we looked at various days of differentiation, the most common motif that was enriched in sites of Brahma occupancy was this REST motif. And REST is a transcription factor that counters neuronal differentiation. And what we find is that indeed, by day 10 of differentiation, where REST is robustly expressed, it's completely absent in the knockouts, indicating that it's an important break for neuronal differentiation. And without it, we can promote, the cells may be prone to promoting neuronal differentiation. And that's in fact what we see if we do siRNA to REST in the wild type cells, is that we get the spontaneous formation of beta-3 tubulin cells. So it's this balance between positive and negative regulators of neuronal and anti-neuronal factors that's important for this de novo formation of neurons in the absence of Brahma. We still don't understand why there's this, the mesoderm makes these neurons, but we're starting to understand what mechanism this takes place by. Okay, so I told you that the mesoderm forms just fine, and one of the things that's really important at the mesoderm stage is this dosage of BMP4. And normally I wouldn't talk about this, except for the fact that when a differentiation fails, we normally play around with the dosage of BMP4. And Sutansu went to extremes with this, whereas normally we'd play within this particular range of BMP4 and have normal and maybe some varying degrees of efficiency of cardiac differentiation. This is what we see here in the wild types. And the Brahma knockouts in these dosages of BMP4, we don't have troponin T staining, we don't have cardiac differentiation. So but Sutansu went and really cranked on the dose of BMP4, and you can see here that if we have a 4x expression or dosage of BMP4, we now completely impair cardiac differentiation. I can tell you now these are now, in the wild type cells with this level of BMP4, are now making hematopoietic stem cells and fibroblasts. But now we've completely rescued the differentiation of the knockouts. And so this impaired differentiation from mesoderm to neurons is completely compensable simply by increasing the dosage of BMP4. Again, we looked at single cell RNA-seq. This is the UMAP of the different genotypes and BMP4 dosages. This is the UMAP of the different cell types, and I'm going to walk you through this. And so what you can see here in the low BMP4 situation, the wild type and the knockouts again cluster together. And now the wild types take this path, so it's cardiac precursors forming this left side of this little heart, and then make cardiac myocytes. And the knockouts, instead, as I showed you, take this path instead and make neuronal precursors. But now, when we crank the level of BMP4, the mesoderm stage is a bit different, but now both the wild types and the knockouts reform the broken heart at the cardiac precursor stage, and now both of them are making cardiac myocytes. The low dose wild type BMP4 and the high dose knockout BMP4, telling us that we've now completely rescued simply by the whole path of differentiation, simply by cranking up the dosage of BMP4. So what I've told you in this vignette is that normally you'd expect in the Waddington landscape dogma that once you get committed to mesoderm, you're locked into mesoderm and you can make cardiac myocytes, or you can make hematopoietic stem cells or fibroblasts, but you can only make mesodermal derivatives. And what I've shown you here is that despite this going down the path of mesodermal differentiation, by removing Brahma, we've now uncovered what theoreticists refer to as a saddle node bifurcation, whereas the path that was normally occult to the genome is now accessible and visible, and whereas this path towards cardiogenesis is no longer accessible to the genome, it gets shunted down to this other path that would normally not be accessible. And we're working with mathematicians to mathematically model this, and based on the experimental data that I've showed you, the mathematical models hold that this is in fact what's happening. And so completely changing the dogma that once you're knocked into mesoderm, that you can no longer ever access these other values. And so I'll stop there. This is a relatively recent modern times depiction of our lab. These are the folks in the lab. I've mentioned the important folks along the way, former members that participated in the project, and then many collaborators. And we wouldn't have been able to do any of this work without our fantastic cores at Gladstone. and then we'll stop here and answer any questions.
Wow, fantastic. That is just an amazing journey. I think it's like a great detective novel that you start out with a crime of a congenital heart disease, and the TBX5 was your clue. And then you really actually figure out the partners, the modus operandi, and also, the three-dimensional relationship, of the various activators and also counterbalancers in this process. It's just sort of mind-boggling how nature can actually do this with every embryo correctly.
Right.
It's really always inspiring. Yeah. So I don't know if there are questions for Dr. Bruneau. I mean, this is just a absolutely mind-expanding lecture. It's just so exciting. Maybe I can start by asking kind of just a high-level question, because you really showed very elegantly in terms of how TBX5, for example, can work with MEF2C, and in terms of determining the balance of downstream responders, and that these and then similarly, in terms of the BAF complexes, Brahma and BMP4, sort of have these delicate balances. So obviously, it's really kind of the local balancing of these factors in the target cells that ultimately determines the fate of these contributors to the cardiac development. So how does the input function, for this balance ultimately gets determined, because, right now you have TBX5, which can kind of forward, determine some of these balancing. Where does it get the feedback? Because obviously you figure this out by the neighboring cells, whereas, like what's the like the GPS system that they get the signal from their neighbor to provide this feedback, and so that you can really actually get everything right at the end of the day.
Yeah, and you're bringing up an important point, is that everything's in a precarious balance, right?
Right.
As you said, it's amazing that 99% of the time, the heart's formed normally, right? It's absolutely incredible. And obviously there's some variance, and there's some variance that isn't necessarily manifest as what's clinically recognized as a congenital heart defect, right? And it's only when you actually do recognize it as such that you say there's a defect. So there's range. And why would there be such a balance, if it's so precarious? My thoughts are is that you, as you're pointing out about the feedback mechanisms, is that you need a very fine regulation of a quantitative regulation of gene expression. And so you need to be able to have what ends up being a robust feedback and feed forward set of loops, but that's very sensitive to dosage, to stoichiometries, because you want to be able to say, in this cell population, I want this level of gap junction, whereas in this, I want this lower level, to be able to have conduction system versus working myocardium right next to one another.
Right.
And so I think that the feedbacks are enacted as the heart develops, and there are cell interactions that we don't quite yet understand. I mean, early on in development, it's broad gradations of signaling across the early embryo, and we're working on trying to understand that, and that gets refined more and more as cardiac precursors get established, and then as morphogenesis takes place. And so one of the things that we want to do, going back to the first set of slides where we're looking at the mouse, and we have this lineage boundary, we want to understand, we know now where to look. We can look at these red cells, and now we're going to say, what's happening in those red cells? What's the signaling mechanism? And one interesting observation that we've made is that if we kill off those cells with a genetic trick that uses diphtheria toxin, we now get a hypoplastic right ventricle, which is not at all what we expected, implying that what you're talking about, the signaling mechanisms, are important very early on, not just for subtle regulation, but for very broad aspects of morphogenesis. So we don't yet understand all of the regulatory loops, but we're beginning to understand them based on this work. So
Yeah, it's absolutely amazing in terms of, so, so a couple points I also kind of got from your lecture is that in fact, there are these unexpected compensatory mechanisms that in fact can be activated, even though, let's say, the path has gotten, onto a wrong start. This is a great illustration in terms of sort of how the BAF, complexes, sort of ultimately can correct itself. But the other thing that was, and I was curious, in terms of your NIH grant as well, but in terms of the data you've shown, in terms of how quickly, after the embryo development, just, from day 2 to day 4, there are just dramatic, right, sort of reversals of these programs. And so obviously, there is a temporal clock that's running at the same time while all this is going on, in addition to the kind of a geographical aspect, which you nicely mapped out, from 2D to 3D to, really to the entire network. So I'm kind of curious, and of course, curious about your grant as well, so how does this temporal, is this, the usual biological clock we're talking about? Or is this a whole, different set of determinants, in terms of the, it's a, mass action of what's happening with all the genetic programs?
Yeah, I mean, so at every cell cycle, the genome gets completely disrupted, chromatin gets completely disrupted, and then comes right back together. And it
Right.
normally comes right back together in the exact same shape. The same connections get made, and that's absolutely fascinating how that takes place. But what happens is that there's a notion that there's some memory of the organization, but then that then gets changed as various transcriptional regulators get turned on, recruit complexes to different sites, and so the topology of the genome changes and the enhancer promoter communications change. And we have data that indicates that in the TBX5 heterozygous cells that there's a dynamic 3D interaction of promoter enhancer loops that's actually in some cases altered, not in terms of impaired, but actually loops that are being formed that shouldn't form in the first place. And so ectopic loop connections that are being made. And we suspect that what that's doing is that it's titrating away the productive loops that are supposed to be there to activate the genome. And so there are things that happen very, very quickly. We've done some in the context of 3D organization with other factors called CTCF and cohesin. We've done single molecule, live single molecule tracking in cells. And the movements of the factors is really fascinating, that there's some transcription factors that move very, very quickly and other molecules that remain very stable in one particular location in the genome for several minutes, not just several seconds, whereas others are sort of sampling the genome until they find a place that they like, and then they sit there and they land there. And so the genome is extremely dynamic, and those productive interactions are, I think, a result of I guess the right factors finding one another for something that becomes a productive interaction at a particular locus.
Wow. So I guess you are, just amazing in terms of what components of the genome are accessible versus not accessible, at different times makes a huge difference. And obviously, these are dictated by these complexes as well. And
yeah, that's what we're trying to understand at the moment is how the accessibility
right.
is regulated. Definitely, we think that the chromatin modelers are involved.
Right. Okay.
I see a question in the chat.
Yes, that's right. Yeah. So Katie, thanks. Thank you for the great talk. And wonder whether you have a look at the interaction of chromatin regulation and the environment. So I guess so the microenvironment and the greater environment, and these enhancer regulator more subtle or possibly controlled during stresses, as an example. Yeah.
Yeah, so stress, for example, let's say in the kind of work that Peter works on, heart failure, hypertension, and those types of things. There's definitely very dynamic regulation of enhancers in that in that work. A colleague of mine, Sap Haldar, showed several years ago in a very elegant set of experiments that bromodomain containing proteins were important for the regulation of enhancers during cardiac hypertrophy, but not exercise. And with the notion that you could drug the enhancers to control cardiac hypertrophy. Turns out that those drugs are toxic, but the concept is there that you can regulate the enhancers and you can prevent or even treat established hypertrophy by modulating the dynamics of the enhancers. It's well known that maternal diabetes affects the predominance of congenital heart disease. And my colleagues next door in Deepak Srivastava's lab are trying to understand how the concepts that I talked about, chromatin accessibility enhancers, are affected by those things. And we're going to be collaborating with a new recruit at Gladstone, Isha Jain, who works on hypoxia and hyperoxia as well as vitamins, to try to understand how maternal diet can interact with the congenital heart disease mutations. Because what we don't understand is the variability of the expressivity of mutation. You can have within the same family, the same mutation, pretty much the same genetic background, a very different type of congenital heart disease. And I don't think that it can be explained solely by genetic background as a whole, that there must be other environmental factors. And so we'd like to understand what those are and maybe be able to control those so that even if you did inherit a particular mutation, that controlling maternal diet might be helpful so that that defect is then less severe than it might be otherwise.
Absolutely. And I always feel very depressed after learning about congenital heart disease patterns, and that fathers don't really actually count very much. It's the mothers and the environment that really, sort of have such influence in terms of the offspring. Maybe I can check to see if there are folks who would like to ask a question directly to Dr. Bruneau, and you can raise your hand or also unmute your microphone. I don't know if Dr. DeBode would like to make a comment. So it'll be certainly a great honor. Maybe the mic is not.
Well,
Oh, yes, go ahead.
oh, I'm sorry. I have a problem with problem with the with the microphone here.
We hear you.
We can actually hear you. Yes, we can actually hear you. Thank you so much.
Well, I don't know. Hello?
Yes, we hear you.
Can you hear me? I'm sorry about this.
Yes, we hear you.
I was so, since you know some of the role of some of the DNA binding proteins in general field of congenital malformation, do you see any role of DNA editing to prevent that kind of a thing?
Yeah, that's a really great question. Yes and no. Right. So one might imagine. So it's already there's IVF screening being done for known mutations. So for example, Holt-Oram syndrome, which is really devastating. There's also very severe limb defects. People are doing IVF genetic screening. So there you don't need to edit, because you basically pick the IVF, the fertilized embryos that don't have the TBX5 mutation. And one wants to be very careful with the notion of editing embryos or editing gametes. But one of the things that's been tremendously promising, this is from my colleague Eric Olson, is to edit somatic cells, especially so in particular in the context of Duchenne's muscular dystrophy, where to cure literally cure. They've done this in dogs, in muscular dystrophy dogs, puppies, with adenovirus that homes to muscle and heart to snip out the mutant exon of the dystrophin gene and completely correct the defect. And so you can imagine doing the same sort of thing for, let's say, an arrhythmia that has a particular mutation. So the transcription factors that I talked about are also implicated in GWAS that are associated with arrhythmias. And so you can imagine doing a similar thing where you could correct in the heart either the binding site in a GWAS hit and do things like that. So I don't think we've figured out the safety issues yet, but that definitely would be a possibility.
Yeah, I've read within my ignorance about lipoprotein metabolism as a target of DNA editing, but I thought that correcting an anatomical defect of an organ that develops so early during development would be I don't know how that would happen. That's why that's a reason or the basis for my question.
Yeah, no, so definitely. So one would need, I mean, as the pediatric cardiologists know better than I do, once you've detected the structural anomaly, it's pretty much too late to correct it. What I would like to think of, and this has to do with sort of the compensability that we talked about, and the dosage of the very sensitive nature of the dosage of the transcription factors, is to be able to discover what it is that modulates that dosage sensitivity, and perhaps, as is the case with folic acid for neural tube defects, have something that is a dietary supplement or something that you should avoid in the diet to just help nudge, because all you need is a slight increase in the robustness of those gene regulatory networks so that things don't fall apart as much as they do. So if we're able to find the thing that will modulate the network just up a nudge, I think that would be a solution. So not genome editing, but in fact something that will just prevent the GRN from falling apart in the first place.
Yeah.
Thank you. Great talk,
Thanks.
by the way.
Thanks.
Yeah, thanks. Yeah. Thanks, Adolfo. And yeah, so just in terms of Benoit's, so in terms of these modulators, it may actually be a tremendous opportunity down the road, in terms of heart failure, which is such a general, challenge from that point of view. And of course, eventually, probably, in a couple of months, you'll be able to reprogram yourselves and be able to put in and plug up those holes, and away we go. Yeah. Are there other questions? Yeah. So Hank Kim, thank you for a great talk. It's quite a crazy loss of BRM completely changed the fate of cell lineage. What could make this unchangeable differentiation compared to other transcription factors? Does it require irreversible chromatic structural change?
Yeah, so Han, you're asking the question that still remains a mystery to us. Peter referred to this as a detective story. We still haven't figured this out. It's a complete mystery, and then it's a mystery of why doesn't this happen in vivo also, right? And part of the reason is because I mentioned the importance of the directed differentiation system. We're keeping the instruction set fixed. We're not giving any wiggle room. And so in vivo, you've got a lot of wiggle room. There's a lot of compensation that takes place. So at least we understand that that's the possibility, and we showed it with altering the BMP4 dosage. Now, why this as opposed to any other transcription factor? I mean, we know from reprogramming experiments that you can force a cell to adopt a completely different fate simply by overexpressing one or two transcription factors, right? It's extremely powerful. So what we think we've uncovered, quite accidentally, is a fragility of a mesodermal state that's unanticipated, that that then leads to basically what we think is a self-reprogramming. We think this is sort of an accident of the experiment that we're doing, and we know that many neuronal genes retain open chromatin, and so I think what all that needs to happen, it seems overly simplistic, for mesoderm to remain robust and to then continue down whatever it is that it's going to give rise to, is to ensure that the mesodermal transcription factors and what they then turn on remains on. And there's a really nice paper that's shown that you can overcome the exit of pluripotency, not committed mesoderm, but you can overcome the exit of pluripotency towards neuronal fate simply by overexpressing any mesodermal transcription factor. This is the work of Sebastian Arnold. And so we think that despite what we think of as extreme robustness, it's actually these cell fates are not as robust as we think they should be. And this is in a sense borne out by long time ago cell transplantation experiments, where if you take a cell from the epiblast and put it in a different place, it's not going to retain its fate. Is going to adopt the fate of its neighboring cells, and so the environment, the signaling environment, will reprogram in that cell so that it knows its place, quite literally. So I think that's what we've done in our admittedly highly artificial system, but we've uncovered this unanticipated plasticity.
Wow. Let me see if other questions. But in view of the hour, I think maybe we will let Dr. Bruneau go. But this has just been an unbelievable scientific journey. But it's really like a detective story. Every chapter, makes it even more interesting. And we are just at the beginning of the book, and I can't wait until we actually find out what the future chapter is going to be reading like. But along the same time, we uncover a lot of the understanding of not only congenital heart disease, but in fact, how cell fate is actually masterminded and how this can be potentially altered, so for therapeutic opportunities down the road. So this has been just, mind expanding journey. It's just fantastic, so thank you so much, Benoit, and we are so proud of your accomplishments, your connection with the Heart Institute, Dr. DeBow joining us today to remind of that connection, and to look forward to opportunities in which we can work with you, because there are many sort of connections from application point of view, and certainly a privilege to have you with us, and hopefully one day in the future we can have you back in person, when all this is over. And I'd like to thank also the Potter family for supporting this very important event, and thank everyone else for joining us today. And so thank you again, Dr. Bruneau, for this treat.
Thank you so much. It's been a distinct honor.
Yeah. Take care, everybody. Yeah.